Hello everyone. Today it's my pleasure to introduce the article. Comparative multiomics analyses revealed a breed effect on the colonic host microbe interactions in pig. Dysregulation of the gut microbiota often leads to immune-related disorders, indigestion, or diarrhea. Here, by using the Jiaxing Black pig, JXB, a local pig breed in China, as a model, we performed comprehensive metagenomic and transcriptome analysis to reveal the interactions between intestinal microbiota genes and gene expression pathways in intestinal tissues. Highlights. First, colonic gene expression signatures of JXB and DLY were highly enriched in metabolic and inflammatory responses. Second, Lactobacillus was the dominant flora in DLY, while Lachnospiraceae was dominant in JXB. In addition, Pacebacteria, Streptophyta, and Aerophobetes were identified as key bacteria associated with PI3K AKT-mediated immune responses. Third, Ig-like domain-containing proteins are involved in regulating gut microbiota metabolism, and ITIH2, PAEP, and TDRD9 are involved in NLR-mediated gut microbiota immune responses. Metagenomic data showed that there was no significant difference in alpha diversity and beta diversity between the two breeds, and the main differences were reflected in the relative abundance ratio of Bacteroidetes to firmicutes, the degree of interaction between gut microbes, and the dominant bacteria. To detect the differences in the transcriptome profiles, RNA-seq was carried out. We further performed an enrichment analysis using KEGG to determine the differences in gene functions. Results suggested the differences of metabolic and inflammatory responses in the colon tissues of these two pig breeds. To identify genes contributing to the differences between the JXB and DLY colons, we conducted weighted gene co-expression network analysis, WGCNA. Ultimately, 452 DEGs were categorized into 12 modules. Module 6 and Module 11 showed the strongest positive correlation with JXB and D pigs, respectively. IQCF5 and RNA-directed DNA polymerase were finally identified as key gene in these two modules. To investigate which microbial taxa are responsible for the differential pathways in the gut of both the JXB and DLY pigs, sparse CCA analysis was carried out with the data of all 10 pigs, the upregulated genes, up group, downregulated genes, down group, and genes whose expression did not change, common group, individually analyzed. According to KEGG enrichment analysis, genes interacting with microbes in the common group were enriched in multiple pathways involved in the immune response and metabolism regulation. The microbes Pacebacteria, Streptophyta and Aerophobetes exhibited interactions with both the upregulated genes and the expressed genes involved in the PI3K ACT signaling pathway and the metabolic pathways. Through metagenomic functional annotation, we discovered that the gut microbiota of Jiaxing Black pigs performs better in NOD-like receptor, NLR, mediated immune responses. In contrast, the enriched pathways in the gut microbiome of DLY pigs are mainly related to metabolism, particularly glycerolipid metabolism. Subsequently, we identified significantly altered subsets of KEGG orthology, KO, in the NLR signaling pathway and glycerolipid metabolism through correlation analysis to find modules with the strongest associations with these KO subsets. Furthermore, key genes within these modules were determined, with Ig-like domain proteins being crucial host genes regulating intestinal microbial metabolic functions in DLY, while ITIH2, PAEP, and TDRD9 primarily participate in modulating NLR-mediated immune responses in JXB gut microbiota. Summary. First, in this study, we present the differences in colonic transcript levels and gut microbiota composition between JXB and DLY. Second, the regulatory effects of gut microbes on the host colonical and, in turn, the host on the gut bacterial immune response and metabolism were determined, and key genes and microbiota were identified. Finally, these findings provide insights into the mechanisms of gut microbiota and their potential applications in animal production.